We'll start off with the basics of DNA. Now, DNA stands for deoxyribonucleic acid. The DNA codes for the sequences of amino acid. So previously you may have already looked at the video on the levels of protein structure. We have the primary, secondary, tertiary and quaternary. And if you can remember, primary is the sequence, is the order of the amino acids, and that particular order, the particular sequence is actually determined by the DNA code. This is the sequence of amino acid, the primary structure, which is going to determine the shape of the protein in our tertiary structure. So if the primary structure's amino acid, if the arrangement is in a different arrangement, it will change the shape. And if you take one step backwards, what can change the primary structure? It is the DNA code. So if the DNA code is changed, then it has an impact on the primary structure. And if the primary structure is changed, and that has an impact on the structure, on the shape of the tertiary structure. And this is the level of understanding and thinking we need to be getting at A level. In here, I have my tertiary structure being explained here, and the reason why we want to go over this, because this is really important for exam. In here you have different structures made out of spaghettis and marshmallows, but what I want you to focus really is that these structures, they are maintained, they are held in position by these spaghettis. So if you were to break the spaghetti, break the bonds, then you're going to change the shape. In the same way, I have my tertiary structure. So let's look at the enzyme here. This is the tertiary structure, and this tertiary structure, this very unique shape, is again held, but not by spaghettis. It's held by bonds. And in here I'm showing you my tertiary structure: ionic bond here, hydrogen bond here, disulfide bond here. You also have hydrophobic interaction. These are the bonds that are holding the tertiary structure. So the tertiary structure is maintained by bonds between the amino acids, such as ionic and hydrogen bond. So if the arrangement of the amino acids are changed, then that means the bonds are going to change. The bonds will break and the tertiary structure will change as well. Our DNA is a polymer. So the monomer that makes the DNA is called a nucleotide. I'm showing you here one nucleotide. That's one monomer. So lots of them joined together forms a polymer. We need to know the structure, what the DNA, what one nucleotide consists of. A nucleotide consists of three things: a phosphate group, that's my phosphate group here, and a pentose sugar. This is my pentose sugar. You can see there's a 5 carbon. Now, it's got 6 carbon actually, but it's the shape is pentose. So it's a sugar, so it's C6 carbon, but the shape of it, as you can see here, it's got 5 sides. 1, 2, 3, 4, 5. It's a pentose sugar. But for A level, we need to know the name of the pentose sugar: deoxyribose. So pentose sugar is a generic name, but the specific name which you have to know for DNA is that pentose sugar is deoxyribose sugar. So that's the specific name of the sugar. And the pentose sugar, or the deoxyribose sugar, is attached to a nitrogenous base, and we have covered this. These are 4 bases: adenine, thymine, guanine and cytosine. Let's go into the polymer. So we said the monomers are called the nucleotides, but when lots of nucleotides join together, we call it polynucleotide. So this is our DNA, but we call it polynucleotide to emphasize that this is a polymer of lots of nucleotides. So how is it made? Now, with all things that we've seen in this module, the condensation reaction is involved in making things. So this is a condensation reaction. Look into here. I have one monomer, one nucleotide. I have another nucleotide. These two nucleotides are going to be joined together by condensation reaction. We need to know between which two groups. There's a bond that will be formed between the phosphate of one nucleotide and the sugar, the deoxyribose sugar of the other nucleotide. And this forms a covalent bond. This forms a covalent bond, a very strong covalent bond. And this covalent bond is known as phosphodiester bond. And that we need to know. As you can see here, this is my polymer. Lots of nucleotides are joined together. And as you can see here, the bond we need to know, memorize this, is between the sugar, pentose sugar, and the phosphate. Sorry, I made a mistake in my spelling here. I have shown you the human spine here, the backbone. The reason because if you look at this one of the polymer here, a strand of DNA, a single strand here, the back of it looks a bit like our backbone. And for this reason, we call this part here, the outside part of the strand of DNA, sugar phosphate backbone. The sugar phosphate backbone is very, very strong. And the reason why it's strong is because it's got lots of phosphodiester bond. And these are our strong covalent bond. And we will see later on why this is important. In here, I'm showing you one of the strand of the polynucleotide. We can see that this is our phosphodiester bond. This is our sugar phosphate backbone. Now DNA is double-stranded. It's two-strand. DNA is always two-strand, and it forms a double helix. So I have one strand here, and this is the other strand here. And we can see that both of these strands, physically, they're not really touching each other, but there's a bond between them. There's a weak hydrogen bond between the bases. So DNA is double-stranded. The two strands are held together by a weak hydrogen bond. It's really important to know that these hydrogen bonds are weak, because later on you will see that in DNA replication, you have to break the two strands separate. You have to separate these two strands. So having these hydrogen bond weak makes it easier to separate and replicate your DNA. So these weak hydrogen bond are between bases. So when the double strand DNA, when it twists, it forms a double helix. The hydrogen bond is between bases and is a complementary basis. This hydrogen bond, weak hydrogen bond, can only form between complementary base pairing. So A and T when I have 2 hydrogen bond, and C and G when I have 3 hydrogen bond. And these dotted lines here are showing you the hydrogen bonds. A and T, adenine and thymine, only 2 hydrogen bonds, and cytosine and guanine 3. We need to know that. So the question is, what is the importance of hydrogen bond between the 2 complementary strands? We have just answered it, but on the last slide we're going to show you all the important functions relating to its structure. So this is the last slide. 5 points we need to know really well. Number 1, we have seen the sugar phosphate backbone. If you can remember, we said the human backbone and the polynucleotide, the DNA strand, the back of it looks like the backbone, so we call it sugar phosphate backbone. And remember, they are held together by a strong covalent bond, and that specific bond was called phosphodiester bond. The reason why they have that, it makes the DNA double helix a very stable structure. We want our DNA to be a very stable structure. This is the most important thing of the cell. Between the two strands, DNA is double-stranded. They're held together by weak hydrogen bond. The reason why this is weak, it allows the unzipping of the two strands, because during DNA replication, the two strands have to unzip, has to separate, and having a weak hydrogen bond allows ease. Complementary base pairing. The two strands, we know that they will only join together in complementary, adenine with thymine, cytosine with guanine. This ensures when the cell is replicating its DNA, because it wants to do mitosis, let's say, it wants to divide. We know that in mitosis, every single cell are going to be identical. So if the complementary base pairing ensures the identical DNA is made in every single cell that is dividing. Large molecule. DNA is a large molecule, and that is very useful for us, because that means we can store lots of information that can be carried out. And later on you will see how that the DNA is wrapped around a histone protein because it's so large and so long. DNA is double-stranded. It's double-stranded, should be, and this allows replication to take place. Later on in your module, you'll cover DNA replication, where both of the strands can be used as a template strand, and having a double-stranded allows you to have your semi-conservative replication.